clap.
Yo.
Hi guys, welcome back to chapter 2 Biochemistry for MCAT, which is enzymes. So today we're gonna learn one short video, what is enzyme and all you need to know about MCAT for enzymes. Okay, so first, what is enzymes? So enzymes are just catalysts which speeds up the reactions. It does not change anything. It does not change delta G, delta H, or the equilibrium position. For example, if there's an uncatalyzed reaction where there is no enzyme, it could be spontaneous. It could still have delta G as negative, but the only thing is it is very slow and it could happen, like for example in 2 years or 3 years. So if you just put enzyme in it, it might happen in 1 second or 2 seconds. So that's where the enzyme make a big difference. Enzymes are generally reusable. So for if you have a equation which is like R plus enzyme gives products plus enzymes, okay? So whatever you put in the reaction, you will always receive it back. So enzymes are not utilized in the reaction. Another thing is it always catalyzes both the forward and the backward reactions together, because if you see this graph, so this is the initial delta G and this is the final delta G, and this is the activation energy or the transition energy state. Okay, so what enzyme does is it just lowers it. So it lowers this big jump in between energies. So still your initial and the final is same. So your delta G is same, or your delta H is also same. The only thing which is changing is this big energy jump. So instead of taking a larger jump, you're taking a smaller jump. So it's taking less time. And then if you go this way, still you're taking a smaller jump. If you're going the opposite way or the reverse way, you're taking the same. So forward and reverse reactions, it does both. Okay, let me just erase all of this. Okay, cool. Let's move to the next part. So what are exergonic reactions? Exergonic reactions means that it releases energy. That means your delta G is negative or spontaneous. Okay. Endergonic reaction means it requires energy. That means your delta G is positive and it is non-spontaneous. Okay, it needs energy. Spontaneous process just happens on its own. It does not need any external support. Okay, then we come to type of enzymes. So we have first one, which is oxidoreductases. Oxidoreductases are used in redox reactions and leads to transfer of electrons. It is important to learn that oxidation means that you have lost the electron or losing of electron. Reduction means gain of electron. Okay. Transferases is it just transfers the molecule between functional groups. For example, adding alcohol group or aldehyde group or carboxylic group. So it just transfers from one functional group to another. So transferases, it's transferring. Hydrolases means wherever you see the last part is lases, that means an enzyme and it breaks something. So that's what's written here. Hydrolases means hydro, that means using water, you are cleaving something. Lases is just cleaving something without using any water, without using any electrons. Okay, so basically lases are used as reverse reactions to synthesize something. For example, if you're breaking, so to synthesize something we use lysases. Then we come to isomerases. Isomerases is just changing a from one isomer to another. So you might have learned in your chemistry classes, isomers is something which is having same molecular formula but different structure, right? So isomerases does something same. The molecular formula remains always same. It's just new covalent bonds here and there formed, which changes the structure and the chemistry. So that's isomerases for you. Ligases, you can learn like ligases are like glues because it has a G in it. So ligases is a glue. So it is used to join two biomolecules together. For example, if there's a cut in a DNA, so to join the two DNA strands together, it's ligases are used. Lipases, so if you see lipases, lipids, it has some connection. So it's used to catalyze hydrolysis of lipids into glycerol and fatty acids. It could be different alcohols, but it's generally glycerol and fatty acids. Okay. Now we come to kinases. Kinase is also another type of enzyme which is used in phosphorylation because it adds a phosphate group. Okay, so phosphorylation is a process we'll learn in further chapters. It results in modifications, post-translational modification, post-transcriptional modifications, those kind of things. So kinases is used to add the phosphate group in something. Okay, so kinase add, and it's also a type of a transferase because it's changing the functional group, right? So kinases is a transferase which adds a phosphate group. Phosphatases is the opposite of kinases, so it removes the phosphate group and it's also a type of transferase. Phosphorylases is also adding the phosphate group, but it only adds to an organic molecule. You will see that mostly in sugars like glucose. Okay, then this is the most high-yield topic of this chapter, enzyme kinetics. Okay, so first let's know what saturation kinetics is. Saturation kinetics is when an enzyme reaches to its saturation point. So what's the basic function of enzyme that I told you? That it increases the rate of reaction. So saturation point of an enzyme is it can't increase a rate at after one point. So for example, if you have 10 enzymes and one substrate, so one enzyme and one substrate will bind to each other and it will release, will increase the speed a little bit, right? Or for example, if you keep increasing the substrate number for say 2, 3, 4, 5, and you have 10 enzymes, so as you increase the number of substrate, the velocity of the reaction increases or the speed of the reaction increases, right? But after 10, you will get a flat line. It won't increase because you have 10 enzymes and all the 10 substrates are now binded to the enzyme. So if you have an 11th substrate, it has no enzyme to bind to it. So the speed will not increase and you would just have a flat line. The best graph to see this is called the Michaelis-Menten graph, which is like a hyperbolic graph. I can show you so this is the graph and it looks somewhat like this, and this is the Vmax point of it. Okay, and if this is the half Vmax point, if we go to the x-axis of this point, we will get the Km. So Km, you see here, Km is the substrate concentration at which the enzyme runs half of its Vmax. So this is half of the Vmax and this is the Km. Okay, you can derive the formula for Km from this reaction which is given to you. So this is enzyme plus substrate at K1. K1 is the speed at which it goes to the enzyme substrate complex, to K2, which gives enzyme plus product. Okay, this part is reversible, so there's K negative one also. This part is generally not reversible. Okay, Vmax you saw is the maximum rate at which an enzyme can catalyze the reaction. This is when all enzymes active sites are saturated with the substrate. As I told you, 10 substrate, 10 enzyme. If you add more substrates, nothing will happen. It's the saturation point of that enzyme. Their graph also gives you an equation that is, so here it is like V naught. I can actually show you. Okay, so this is the Michaelis-Menten curve. So you have V naught on the y-axis and substrate concentration on the x-axis. And so at if you want to find the velocity of the reaction at any point, you will just take the Vmax multiplied with substrate, and then it will be Km plus the substrate. The equation is wrong over here. This is the correct equation. So Vmax S over S plus Km. Okay. Another thing is cooperative enzymes. Cooperative enzymes, the most unique part of it is it does not follow the Michaelis-Menten equations. Okay, as Michaelis-Menten equation you see is an hyperbolic curve. Cooperative enzymes are generally sigmoidal curve, like this. So this is the Michaelis-Menten equation, but which is good for the non-cooperative enzymes, but cooperative enzymes have this kind of a sigmoidal pattern. Okay, then we come to mechanisms of enzyme activity. So any enzyme, for say this is your enzyme, so any enzyme will have like an active site and an allosteric site. So active site is the main site where catalysis happens. Some substrates like to bind to the allosteric site and do a conformational change. We'll see it in the later part of the chapter, but for now active site is just a site where the substrate can bind and and make the enzyme active. Okay, so there are two theories which explains it. One is the lock and key theory. So for like, as you know, every lock has a unique key to it, which will go inside it. So similarly, they say that substrate is a key and enzyme is a lock, and it will have a specific thing that only that substrate can bind to. It will have a specific shape of the active site that only that substrate can bind to. So that is lock and key theory. Another theory is the induced fit theory. Induced fit theory means that a substrate will first come near to the active site and try to bind it, which will cause a conformational change in shape or some bonds, which will result in that substrate fully binding to the enzymes. Okay, makes sense? Then another two things is cofactors or coenzymes. As we learned in the last chapter, the prosthetic groups. So cofactors and coenzymes are generally the prosthetic groups to the enzymes. Cofactors are the metal cations, for example, iron, magnesium, those kind of things. Iron, you can say in hemoglobin, that's a cofactor for the iron, for the hemoglobin. Coenzymes are the organic molecules similarly required for the enzymes. Enzymes are generally protein in nature, so enzymes are the conjugated proteins, and these are the prosthetic groups to it. Okay, just simplifying the terminologies for you guys. Okay, let's clean up the page for us where we can study more. Okay, did we clear this part? Yeah. Now coming to effects of local conditions on enzymes. So if you remember from chapter 1, we discussed how temperature, pH, and salinity affects the protein. Similarly, it will affect the enzyme. So if we increase the temperature a lot, the protein will just denature, or the enzyme will just denature because it's a protein. If we decrease the pH and put a lot of H+ ions or acids, again, the enzyme will just denature. Similarly, salinity, if we put a lot of salts into it, it will denature. And the structures that could denature is the secondary, tertiary, and the quaternary structures, the 3D shapes, the hydrophobic interactions, the hydrogen bonding. Those things can get broken with these three things. It's harder to break the primary structures because primary structures are made up of peptide bonds, which is covalent in nature, and also they're very rigid because of the resonance. They have double bond character, like I showed you in the first chapter. You can go and see, I showed you with these that it can just come over here, and there you can have a negative sign over here, right? So those kind of things. Okay, going back. So this we did. Now we come to the regulation of enzymes. First is feedback inhibition. Feedback inhibition is regulation of enzyme is basically how enzymes are inhibited. So feedback regulation is, for example, you have A plus B giving you C because of enzyme 1. So if you have a higher concentration of C, like you have a lot of C in your body, so what it will do is it will restrict the substrate binding to enzyme 1, which will inactivate the enzyme 1, and then A plus B would not be able to produce C, and eventually the concentration of C will decrease. So that's called feedback inhibition. It's generally seen in hormones more, like if you have a significant level of one hormone, then that hormone will kind of try to restrict those enzymes to produce more of it. Okay. Reversible inhibition is like where for some time that substrate will inhibit that enzyme, and later when the situations are complementary for that enzyme to be active again, it can become active. So it's reversible. Okay. The textbook definition is, you don't need to know it, but I'll just read: the ability to replace the inhibitor with a compound of greater affinity, or to remove it using mild or lab treatments. Okay. So reversible inhibition are of 4 types, as you can see. Where's my cursor? Yeah. So it's competitive, uncompetitive, non-competitive, and mixed. Okay. Competitive inhibition is just where the inhibitor would fight with the substrate to bind to the active site of the enzyme and block it, basically. Okay, so here the major part you have to know is competitive inhibition. Inhibitor will fight the substrate to bind to the active site and restrict it. So the Km value increases, but the Vmax is unchanged. It just means because the inhibitor is fighting the substrate, that means now you need more substrate to bind to the enzyme so that Vmax can remain unchanged. Okay, we'll see it with the Lineweaver graph here, Lineweaver-Burke graphs. In a while. Then we come to uncompetitive inhibitors. So competitive inhibitors were fighting with the substrate. Uncompetitive inhibitors will let the substrate bind to the enzyme successfully, and then it will fight the whole enzyme substrate complex, and will reduce the Vmax because and the Km both, because it's not fighting the substrate, so it will decrease the substrate and Vmax. Then we come to the non-competitive inhibitor. Non-competitive inhibitor is when the inhibitor will fight the substrate and the enzyme substrate complex with equal affinity. So the Vmax will decrease, but the Km is unchanged. Okay. Mixed inhibition is where inhibitor binds unequal, so these both are kind of same. Non-competitive is equal affinity. Mixed inhibitor is unequal affinity to fight the enzyme or the enzyme complex. Vmax decreases, but the Km could increase or decrease depending on the inhibitor. Okay, because it's a mixed one. If it's like fighting more with substrate and less with the enzyme substrate, then the Km will be positive. But if it is fighting more with the ES and less with the enzyme, more with the enzyme substrate and less with the substrate, then the Km will decrease. So basically, competitive and uncompetitive situations. Okay. Now let's see it on the graph. So basically, these are the Lineweaver-Burke plots. So here you see the competitive inhibition. So blue is the no inhibitor or the control. So if there is an inhibitor, then your Km is increasing. So this point is this point is increasing, and then the Vmax is being the same. Here, this is the, sorry, okay, because this point is showing you 1 over Vmax. Now, this thing is the uncompetitive graph. Uncompetitive graph is showing you the Km is decreased and also the Vmax is decreased, okay? Here, non-competitive is the Km is same, but Vmax is decreased, okay? A good way to remember this is that if you draw a line over here, it is kind of an N shape, right? So it's a non-competitive, and if you do like this here, so it's kind of a U, so it's an uncompetitive, and competitive is like you're kind of fighting with somebody, so it's like a cross. That means fighting. Okay, so I'll just write over here, uncompetitive is cross, competitive is cross, uncompetitive is U, and non-competitive is an N. Okay. Irreversible inhibition just means that you can't reverse it. Once the enzyme binds to the inhibitor, then it's degraded for like a prolonged period, or permanently it's gone. Suicide inhibitor is like it's kind of a substrate that binds to the enzyme, but it binds irreversibly, so it's kind of a death for the enzyme and for the substrate both. So it just makes it, and it is done via covalent bond. So covalent bonds are hard to break, as I told you. Allosteric effectors is like if you have an enzyme, so this is your enzyme, this is your active site. So another site where substrates can bind is called the allosteric site. So allosteric effectors are the ones that bind to this allosteric site. It could be positive effectors or negative. Positive is like which increases the activity. Negative is which decreases the activity. And also all the allosteric effectors show cooperativity, so it does not obey Michaelis-Menten kinetics. Okay, let's clean that now. Homotrophic effector is an allosteric regulator that is also a substrate. For example, oxygen is a homotropic allosteric regulator for hemoglobin. So oxygen binds to the active site also and to the allosteric site also to make differences. So it functions at both sites. Heterotropic effectors are the ones that are different than substrate. For example, if this is O2, the one that binds to the allosteric effector will be some NO2 or something else, okay? It won't be same. Then we came to the phosphorylation, as I promised. Phosphorylation is the modification by adding a covalent bond with the phosphate. So if we phosphorylate something by adding a phosphate group during catabolism, that is breaking of energy, it makes the enzyme active. And if it's during anabolism, that is combining the two biomolecules, then it will make the enzyme inactive. Okay. Then there's another modification, that is glycosylation, which is a covalent modification with carbohydrates. So if you add any carbohydrate, it's called the glycosylation modification. Okay. Zymogens are just a precursor to enzyme. If you have heard about trypsinogen, pepsinogen, so it's the inactive form of that enzyme. If something attaches to it, like a cofactor, coenzyme, or something happens to it, then it just becomes active. And for example, pepsinogen, when it comes to a lower pH in our stomach, it becomes pepsin and then works on our food to digest proteins. Okay. And this we went over. This is just the reaction scheme, that enzyme plus substrate. K1 is the reaction rate where it's becoming enzyme substrate, and K2 is the reaction rate, it's E and P. And if enzyme substrate does not become the product, it would just go back to enzyme and substrate at K minus 1. Okay. Another important thing that these sheets are missing are the two terms. So the two terms are, I'll just write over here. So two terms are holo enzyme or whole enzyme, and then apo enzyme. So holo enzyme is the, let me just see. Okay, so holo enzyme means whole, whole enzyme means whole of the enzyme. So it is the deactivated enzyme with a cofactor or coenzyme, and then apoenzyme is just deactivated enzyme that is lacking this cofactor. Okay, so these are the terms that just comes in the passage and you should know. And yeah, that was all for this chapter. Hope you guys have a good day and a good exam. Don't cram the things up, try to understand it. And if you have any questions, just put it in the comment section. I'll be happy to answer. Thank you. Have a nice day.